Diagnostic Microbiology, a laboratory manual. Cerebrospinal fluid analysis. Purpose: to aid in the diagnosis of meningitis, a central nervous system infection, by performing white BC count. Meningitis is a medical emergency. Anatomy: meninges are the three layers outlining the brain for protection. DAB. Dura mater, arachnoid, and pia mater. CSF sampling. Lumpar puncture into L34 or L45, taking a volume between 5 and 10 mL. Distribute equally into 3 tubes. Tube 1 for chemistry and serology, Tube 2 for microbiology, and Tube 3 for hematology. Tube 1 least affected by hemolysis. Tube 2 largely contains the actual infectious agents. Tube 3 least cells from traumatic tab. CSF tests. Tube 1 for measuring total protein and glucose, and sometimes electrophoresis. Tube 2 preserve at 20 Celsius for culture, gram stain, and sometimes special cultures for fungal, MTB, and viral. Tube 3 preserve at 4 Celsius for cell count and cytology. CSF sample rejection. CSF sample can be rejected in any of the following. However, alert physician before discarding. Quality of CSF sample. Hemolyzed CSF caused by traumatic tab or hemorrhage. Traumatic tab is viewed as gradually diminishing red color across the 3 tubes, or clot formation due to plasma fibrinogen. Hemorrhage is viewed as evenly distributed red color across the 3 tubes, or the absence of clot. Physical examination of CSF. Appearance. Normally, CSF is crystal clear, or bloody due to increased RBCs, or turbid due to increased white BCs, or xanthochromic, yellow, pink, or orange due to hemoglobin or bilirubin, carotene, protein, or melanin, or with pellicle formation due to tubercular meningitis. CSF. Tube 3. Hematology. The results of the 3 tubes are interpreted together. CSF. White BC count. One hemocytometer contains 2 chambers. One chamber contains 9 grids. The depth of a chamber is 0.1 millimeter. CSF. White BC count. Formula. White BC count equals CD over V, which equals total count times dilution over number of grids counted times grid volume. Count grids 1, 3, 5, 8, and 9. A grid volume equals length times width times height. 1 millimeter times 1 millimeter times 0.1 millimeter. CSF. White BC count. Dilution. CSF can be counted undiluted, or 1 to 10, or 1 to 20. Dilution depends on wet mount assessment. The higher the white BCs, the higher the dilution. Or CSF appearance. The higher the turbidity, the higher the dilution. In a clean test tube, mix the appropriate amounts of Turk solution and CSF according to the dilution shown here. Mix well and load into the chamber. Calculations. Example 1. Calculations. Example 2. Calculations. Example 3. CSF, white BC count, automation. Hematology cell counters offer CSF counting option. However, manual cell count and microscopic cytology remain the gold standard. CSF differential count, staining. Any Romanovsky stain can be used. Right Gimza, May Grunwald, or May Grunwald Gimza. Spread 20 microliters of CSF sediment on a clean slide. Stain according to protocol. CSF differential count. Viral meningitis. Evidenced by the presence of lymphocytes, plasma cells, and monocytes. CSF differential count. Activated monocytes. Eosinophilic meningitis, indicating parasitic infection. Lymphocytes and plasma cells. CSF differential count. Bacterial meningitis. Comparison between May Grunwald Gimza stain and Gram stain. In both stains, neutrophils and intracellular bacteria are demonstrated. CSF, differential diagnosis. Interpretation. Compared to normal values, bacterial meningitis is characterized by turbid appearance, increased white BCs, predominantly neutrophils, low glucose levels, and high protein levels. While viral meningitis is characterized by normally clear appearance, increased white BCs, predominantly lymphocytes and monocytes, normal glucose levels, and increased protein levels. Troubleshoots. See the following table for possible troubleshoots. Use the following form to report results. References.